Yeah, but this was like a big bet.
Okay, so in this session, we have 3 speakers. It's going to be an interesting sort of direction from Brian will introduce in a second, who is using data to actually learn things about, actually using data for biological purposes and research. We're going to have a middle talk sort of on visualization, and then Salford at the end will give an overview of it all. I think these 3 talks will fit together very nicely. Each talk will be 20. We'll do questions at the end of each talk. So let me start by introducing Brian White. He is joining us remotely from Jackson Labs today. He is a senior computational scientist, where he focuses on the analysis of spatial and imaging data to elucidate cellular interactions within the tumor microenvironment. I think it's sort of interesting. He's also coming from a PhD in computer engineering, who switched into computational biology during a postdoc. So it's nice for me personally to see the number of people who have done this sort of transition. Anyway, take it away, Brian.
Thank you, Eric. I really apologize that I can't join you folks in person, but I appreciate the organizers inviting me and having this chance to share some of our work with you. My title is a mouthful. Let me try to interpret some of it for you. This will be, like Eric just said, an application center talk, biology, where we're using spatial technologies and imaging technologies to profile triple-negative breast cancer patients, and in particular to see how mutations in the BRCA1 protein lead to disorganization, or maybe don't, of cells, in particular cells within the so-called tumor microenvironment, that is, cells surrounding tumor cells. So BRCA1 and or BRCA2 proteins deficiency occurs in about 50% of triple-negative breast cancers, or TNBCs. This can be through a combination of germline variation, acquired somatic mutation, or methylation. Both proteins act in the homologous DNA repair pathway, which is a mechanism for repairing double-stranded DNA breaks. As such, patients with BRCA alterations suffer deficiencies in homologous repair, and this can lead to accumulation of mutations and genomic scars, including chromosomal rearrangements, single base substitutions, and small deletions. And prior work by some of my colleagues in the EDLU lab at the Jackson Lab has shown that BRCA1 knockout, that is a mouse model that doesn't have the BRCA1 gene, recapitulates many of these genomic scars, including the so-called tandem duplicator 1 phenotype that they characterized. But beyond these genomic scars, some studies suggest that BRCA1 deficiency is associated with increased immune infiltration within the tumor, including expansion of the CD8 T cell population. Yet other studies showed no changes, and so the relationship between these so-called TILs, or tumor-infiltrating lymphocytes, and homologous repair deficiency remains unresolved. So as such, we set out to elucidate the tumor microenvironmental response to BRCA1 deficiency in triple-negative breast cancer by profiling naive, that is untreated, BRCA1 proficient and deficient tumors in human and in the mouse model I just very briefly sketched. And we did so using two primary spatial technologies: Codex for spatial proteomics and Visium for spatial transcriptomics. I'm hoping actually that you folks won't be too familiar with these, and part of the intent of my talk is to introduce you to these modalities, and I'll do throughout. And then these were complemented by more standard assays like single nucleus and bulk RNA sequencing, as well as histological assessment of H&E, or hematoxylin, and eosin slides. This is our final cohort. It was comprised of 3 human BRCA1 wild type samples, that is, samples without mutations, and then 7 deficient tumors from a larger cohort. And our mouse sample was a bit smaller and included just 1 BRCA1 wild type and 2 mutants. And for both species, we had to exclude a couple of samples that were characterized by a pathologist as having unusual histology. So to assess whether TILs, those tumor-infiltrating lymphocytes, are indeed increased in BRCA1 deficient tumors, as is our hypothesis, we first turned to Codex. Codex provides cellular and subcellular resolution of 50 or more proteins. The assay, as we're using it, is performed on this phenocycler fusion instrument from Akoya Biosystems. It uses an initial staining with oligonucleotide conjugated antibodies, so antibodies that bind to the protein, and then it's followed by successive rounds of hybridization with a nuclear fluorescent stain, that is DAPI, and 3 reporter oligos. And these oligos are conjugated to fluorophores that are complementary to those conjugated to the antibodies, and that can be read out. So after each cycle, a fluorescent image is acquired, and the fluorophores are dehybridized by gentle washing in preparation for the next stage. And then finally, each of the cycles of 3 channels plus the nuclear stain are registered using that nuclear stain that's common across them. And output is a high-dimensional image in the proprietary QP-TIFF format. As this is a session on more and faster and better, I want to emphasize the scale of the data. So we generated nearly 50 gigabytes of data, at least these primary data sources, the codecs. And so this is a 200,000-foot view of a human BRCA1 mutant with all 38 markers simultaneously illuminated. That is all 38 proteins. And the mouse data includes 35 proteins, many of them which overlap those in the human cohort for consistency. So this is a little more detail. Here I'm showing the size in bytes and pixels of the codex data, as well as the accompanying H&E. The codex QP-TIFF format is a TIFF-like format, and it's pyramidal. Czar and Dask were really instrumental enabling technologies here for our work. And through them, we were able to efficiently ingest in this case, the 6.5 gigabyte slide, the image that I showed in the previous slide, into Napari, essentially instantaneously, as you would expect, in under 200 milliseconds. Whereas a naive approach, which I first attempted, of loading the entire image would have taken 6 minutes. Storage of these large data was also made more cost-effective because of several properties of the PhenoCycler, that is the codex instrument, and this proprietary QP-TIFF format. So most obviously, and maybe not as interestingly, the QP-TIFF uses LZW, lossless compression. But beyond that, the PhenoCycler attempts to optimize the signal to noise by truncating and then dilating the native 18-bit data into the 8-bit representation in the QP-TIFF. And this does have some unfortunate analysis consequences. So it's susceptible to hotspots, setting the high end of the dynamic range, and that's pushing potentially meaningful biological data, not necessarily into the noise floor, but kind of dangerously close to it. And I'm trying to show you an example of that here using the PANS cytokeratine, sometimes they'll call it PAN-CK marker. So this is a marker for epithelial cells, and this is an epithelial tumor. So generally we use that as a tumor cell marker, but inevitably there are some normal cells, normal epithelial cells within these samples. And so here I'm showing some normal duct, the epithelial cells within that normal duct, and they're staining for PAN-CK, and it's very high. You may be able to make out this little blip at the high end of this expression density profile. So this is setting the high end of the scale. And if we were to look within this same sample at that same contrast level at tumor cells, so we've identified tumor cells using an additional marker, CD49F here in red on the left. This is the same image using a different marker that PAN-CK I've been talking about in the center. You can see that the tumor cells are almost indistinguishable. And it's not until we turn the contrast way up that we can finally see via PAN-CK the tumor cells that we know to be there. So this leads to a dramatic shift in the tumor cell expression profile of PAN-CK relative to other samples. So here's the sample I've been focusing on in this teal, JP6346. Instead, here's a representative sample, JP1330, which has a more typical distribution profile of PAN-CK and tumor cells, so shifted well to the right. So this led us to analyze each of the samples independently. As a first step to quantifying the cells, we segmented DAPI and select membrane markers into over 4 million cells project-wide, with 200,000 or more cells per sample. And we used the popular Mesmer method for cell segmentation, which is originally developed for small regions of interest. My colleagues Antonis Samarakis and Fernando Cervantes adapted the approach so that it would scale to the whole slide images that we generated. We next hierarchically classified the segmented cells into using canonical markers. So for example, we first partitioned the cells into tumor cells and then non-malignant or non-tumor cells. We then subdivided the non-tumor cells into stromal and epithelial, and et cetera. And at each stage, we overclustered the cells based on relevant subset of markers and then collapsed the related clusters. And I'm trying to show that here on the right. Now, each of these columns is a cluster of cells, And you can see that they aggregate into metaclusters, so themselves are clustered. So in this case, there are 3 clusters. There's the B cells that are indicated by the phenotype bar at the top here. There are the T cell clusters that span the B cells. And then finally, there's a myeloid cell cluster on the right. And these are based on the marker proteins for these immune populations that are shown on the right. And notice that these markers themselves cluster according to their associated cell types. Ultimately, we characterize the immune cells into myeloid cells, B cells, and CD8, CD4, and regulatory T cells, along with stromal and tumor cells. And here you can see the relative expression profile of the protein markers for each of the cell types. And you can notice that the T cells here cluster together, and they express the markers that we would expect, the pan-immune CD45, as well as T cell specific markers CD2 and CD3 up here. And then myeloid cells, just as one other example, they also express the pan-immune CD45 being themselves immune cells, as well as the myeloid-specific markers CD68 and CD11B and C. So we can now finally view the classified cells spatially. The tumor nests that are clearly demarcated by CD49F on the left are classified as tumor cells in cyan on the right. And then the cells between express CD3, so these are T cells, and they're characterized on the right into several flavors of those T cells in yellow, red, and purple. Now, our hypothesis, remember, was that these TILs, the tumor-infiltrating lymphocytes, are increased in the BRCA1 mutants. So as a final step, we need to place the phenotype cells into their tissue morphological context in order to distinguish between lymphocytes within the tumor, tumoral lymphocytes, and stromal lymphocytes, those that are outside of the tumor region. So to do this, we leveraged a protocol innovation from our colleagues at Jackson, the single-cell biology lab that generated the data that enables a single tissue section to be profiled molecularly with codex and histomorphologically with H&E. We then registered the 2 images to achieve a single cell alignment of the 2 modalities. And this movie will fade back and forth between the codex signal that identifies the T and tumoral cells according to their markers and the H&E image that shows the connective tissue surrounding the tumor nests. This movie and all the spatial data that I'm showing were generated using the Tapari viewer. And it's our colleague, Peter Sobolewski, who's a co-developer of this platform, really helped us and guided us in applying it to our data. Our pathologist collaborators then annotated the H&E images into stromal, extratumoral lymphocyte, and invasive tumor regions. And then having aligned both the H&E and these annotations to the codex data, we can finally distinguish between the stromal lymphocytes that I'm showing in the top left. These are confined, in this case, to the yellow annotated extratumoral lymphocyte region, and the TILs that we care about on the bottom left, and these are within the annotated blue tumor region. Now, surprisingly, after all of that, we observed that the frequency of the lymphocytes within the annotated tumor region did not differ by BRCA status in either the human or the mouse. Nevertheless, what was striking was that there's a high proportion of myeloid cells at about 20% in both species across most of the conditions, the possible exception of the mutant mice. And these myeloid cells, we notice, often aggregate around tumor nests, and I'm showing that here across both species and independent of BRCA status. So as I've used before, the cyan CD49F marker is indicating the tumor cells in both species. And then I'm indicating the myeloid cells in red, and I'm using a different marker on the left in human, that's CD68, and on the right in the mouse for MHC class 2. But hopefully you can clearly see that there's a lot of red surrounding the cyan tumor cells. So we went on to quantify the proportion of each cell type surrounding those tumor or epithelial cell aggregates at distances of 5, 10, and 20 microns from the periphery of the aggregates. And then consistent with the images that I just showed you, we found that about 20% or more of the cells surrounding the epithelial cells were myeloid cells. And you're seeing that as the blue concentric rings. So this indicates that, as we kind of saw anecdotally, that the myeloid cells frequently surround epithelial cells, at least when those aggregates do indeed occur. And then we wanted to show that the aggregates themselves occur frequently. So we began by defining neighborhoods of a central cell by counting the cells, the cell types within a radius around that central neighborhood. So as an example here, I'm showing a central tumor cell in pink purple, and you can see that within its neighborhood indicated by this dashed line here, that there are a lot of other tumor cells, as well as a lot of myeloid cells. And so we can just count these up and we get this expression profile for each cell. And then we can cluster these expression profiles across cells to define communities. And here I'm showing you some of the most prevalent communities in both human on the left and mouse on the right. So the communities are the rows, and the cell types constituting those communities are the columns. And what we can see is there are epithelial myeloid, that is tumor cell myeloid communities in both human and in mouse. I've indicated those in the box. And they're composed almost entirely of these two cell types. So in human, that community has about 50% epithelial cells and about 40% myeloid, and the mouse community is similar. So finally, we quantified the proportion of cells belonging to each of these communities. And we observed that about, and I'm showing that proportion in sky blue here at the top of the stacked bar plots. And we see that about 10% of the cells in most of the samples belong to that community. So we thus conclude that this arrangement of cells is prevalent in both species and across both genotypes. So to elucidate the pathways, that's kind of the downstream consequences that might be activated when myeloid cells and tumor cells are juxtaposed within these communities, we next turn to a different modality, Visium. This is a spatial transcriptomic platform that profiles the whole transcriptome, that is all the genes in the genome or the transcriptome, and it does so within 50 micron spots. These are arrayed across the tissue, as you're seeing here in this cartoon, but they're separated themselves by 100 microns. So when we apply it, we see broad expression of the epithelial marker keratin 5, as we would expect, and the myeloid cell marker CD68 in yellow. And then greenish spots express both keratin 5 and CD68. And additionally, as with the Codex data, we generated a high-resolution H&E image of the same tissue section. Zooming in, we can see how the 55 micron spots capture about 10 to 20 cells after I've segmented the cells. And we see that there are some strongly blue cells that presumably contain mostly epithelial cells, and strongly yellow cells that contain presumably mostly myeloid cells, and then those greenish spots that contain a mix. But we can quantify this, we can make this more precise by estimating the cell type fraction in each spot using so-called deconvolution methods that I'll describe briefly here. So to do so, we performed single nucleus RNA sequencing on 2 samples for each genotype in both species. And then my colleague Tausif classified these cells, which I'm showing here, demonstrating that we captured the major immune and stromal and tumor lineages across both species. Deconvolution then defines an average expression profile for each cell type by aggregating and averaging the classified single cells. It then seeks a weighted sum of those average expression profiles that best approximates the spot's observed expression profile. And those weights are effectively then the proportion of each cell type. Tausif's deconvolution on the right is highly concordant with the marker expression profile on the left. The advantages, though, of the deconvolution are that it smooths the noisy low count expression data, and it puts them on a scale, namely the proportions, that are interpretable and comparable across cell types. So if we look into one of these green spots that we noted previously that we speculated had both myeloid and epithelial cells, we can indeed see that and quantify that. So this greenish spot has maybe 60% tumor cells and maybe 15 to 20% myeloid cells. We went on to validate these deconvolved cell type proportions by comparing them to Codex-derived results on a near adjacent tissue section. So as I mentioned previously, and I'm showing here again, both modalities, the Visium and the Codex, are paired with H&E performed on the same tissue section. So we aligned the associated near adjacent H&E images from Codex and from Visium, and then we applied that alignment to the associated molecular data, so the Visium transcriptomic data and the Codex data. And so what I'm showing here at the bottom right is the Visium data after it's been aligned to the codex. And so hopefully you can see, so we use the H&E as a backbone, and then the molecular data comes along for the ride, and they're kind of nicely overlapped, as we can see here. So we generally see broad consistency between the two modalities in terms of the cell types and the markers they express. For example, the pockets of yellow CD45 shown in the codex data on the left correspond correctly to high immune cell fractions, also shown in yellow from the deconvolved Visium data on the right. And these are surrounded by, these pockets of yellow immune cells are surrounded by blue epithelium, as indicated by pan-CK, pan-cytokeratin codex, and the high epithelial cell fractions from the deconvolution Visium on the right. So a little more of the quantification there. We overlaid a 4x4 grid on the data, as I just showed you on the previous slide. And then within each region, compared the cell type fractions derived from codex, I'm showing that on the x-axis, with the deconvolved fraction from Visium on the y-axis. And we see high and significant correlation across all but 2 of the 16 regions. So we then went on to do the analysis that we actually cared about, looking at downstream pathways and consequences. So we thresholded the deconvolved proportions to indicate the presence or absence of each of the 2 cell types that we cared about. In particular, we classified spots into 3 groups based on whether or not they had both myeloid and tumor cells only, or myeloid cells only. And then my colleague, Tao Schiff, calculated gene set enrichment scores, so measure of an activity of a pathway across about 300 reported pathways to see how they differed across these cell types. And we found that a particular glycolysis gene set, which was defined from myeloid-specific cells, was enriched in these epithelial myeloid spots relative to either epithelial only or myeloid only. And we reasoned that pathways exhibiting this pattern reflect an interaction between the myeloid and epithelial cells, as opposed to a simple independent contribution from one cell type or the other. And this suggests that there's a coordinated set of metabolic and cellular changes within macrophages or within myeloid cells that switch them towards a more pro-inflammatory state that's represented by this glycolysis gene set. And that's specific to their being in the presence of tumor cells. And Telsif found that this trend generally held across samples. In addition to this glycolysis gene set, we found others related to myeloid cells and macrophages, were a specific type of myeloid cells, were also specifically upregulated within these myeloid epithelial cells. And this is something we intend to pursue. So to summarize our biological conclusions, we showed that myeloid cells are abundant and surround tumor cells in human triple-negative breast cancer, and that these findings were recapitulated in a mouse model of triple-negative breast cancer. And finally, I just showed that several pathways, including glycolysis, are specifically upregulated in regions that are rich for both myeloid and tumor cells. From a more technical perspective, we generated 50 gigabytes of highly multiplex spatial proteomic data with codex across greater than 35 markers and across 13 samples. And then we aligned these data with spatial transcriptomics data, as well as histological data in the H&Es and other derived data, including segmentation masks and classified cell masks. And finally, we scalably and interactively visualize these data by using Dask and Zara to ingest them efficiently into Napari. So I'd like to close out by expressing my gratitude to my colleagues at JAX and our analysis team for shaping and executing our strategy. Our work was funded internally by the JAX Cancer Center, and it was truly a JAX-wide collaboration involving faculty labs, as well as a number of different departments, including the single-cell biology lab that generated the data, histology, and importantly, research, IT, and imaging applications. This is Eric Radamiro's team. And I want to call out specifically, again, Peter Sobolewski for really introducing us to Napari, which is a major enabling technology for our work. And if I've left time for questions, I hope I have. I'd be happy to take some.
Yeah, thank you very much. We have time for 1 or 2 questions. Okay, first of all, I think it's great to see the rest of Eric's team pop up on some of these slides. Thanks for the application. Okay, we have Jason.
I mean, it's great, and it's really important work too. What would have made things better? So if ZAR were better, or, you know, what would be better for you to have made your work easier, faster?
I specifically belabored this nonsense about the normalization for this crowd. And I didn't go into all the intricacies, but it's really been a huge headache. Essentially, each slide is on its own scale, and it's not clear to me what that scale is. That's a black box normalization. So it's very difficult for me to compare across slides, across patients. So what does that have to do with you folks? That, my understanding, that was done to limit the size of the data, right? From a native 16-bit to 8-bit. So had we had use of the full range of the data, that would not have been an issue for me. So you know, if there's anything there for you folks, I don't know. You know, maybe it's just that this data should have been housed in the cloud as opposed to on our HPC, and that would have given me more freedom to store larger data. I don't know, but that's been a major headache for me. I don't know if that's helpful to you.
No, that's great. No, that makes sense. Absolutely. Thank you.
But yeah, thank you very much. Oh, I just one little note on that bit about the depth of data. Yeah, it seems like in this world where with microscopes and things, my world was 12-bit data, but it wasn't really 12-bit. You want to weigh 60 bits on it, et cetera. So I think this applies to many different domains. Yeah, you're very well. Well, thank you very much. Oh, one more quick question.
Note that Akuya may have seen that because their current QB TIFFs are 16-bit.
Well, that's great. Doesn't help me. Actually, I'm told that we can go back and on the instrument, we have a raw form of the data. I haven't yet been able to convince my colleagues to make that available to me. But I'm glad they've made that change.
Thank you very much.
Thank you.